Everybody for showing up on a Friday, such a sunny day here in Copenhagen. So the title of my talk is rather generic and underlies the general scope of the group that is, right? So, and as I said, control is reliant on a profound understanding of the process. Pretty much, we are very well aware that the general opinion are controlled by social media. It turns out, as I will show you, but by the time you understand at the molecular level how proteins or how chemical reactions work, it can have a profound implication on controlling a chemical output or a biological output at the protein level, at the cellular, and even at the organism. And I'm part of the Department of Chemistry, of course, and the Novo Nordisk Center for Protein Research, and also in the Innsbruck cluster for integrated structural biology at the University of Copenhagen. So I will make a small introduction to the journey across space, time, and that I have followed, and my group has followed over the last years. I started at University of Crete. I did my bachelor in chemistry and my master's and PhD in chemistry, but working with proteins under the supervision of Iulias Monu and also with the help of Michael Orfanopoulos. Then at this time, I started working with proteins, and this is a picture of the University of Crete, and you can see that you can really struggle to get a cloud over there during the winter. So then I moved to the University of Nijmegen under, in the group of Ronald Nolte and Alan Rowan. And at this time, we set up some single molecule experiments in collaboration with Johan Hofkins in Leuven in Belgium. And at this time, I was very happy to meet Thomas Bjornholm. We were in the same European network, an FP7 at this time. And Thomas was starting as the director of the Nanoscience Center, and he said, you should come here. I met and I worked in the group of Dimitrios Stamou for many years. And since mid to the end of 2015, I became independent. And with a great interaction and help of Knud Jensen, I established my own lab away from the department of from the physical space of the Department of Chemistry, but also associated, of course, with chemistry. And here, this is here in Frederiksberg. So, as I said, I'm a chemist by education. My first bachelor thesis was on mechanism of chemical reactions, oxidation of alcohols by dimethyl dioxyrane. And it was at this time that I started interacting more and more with proteins. I was very inspired by a talk of Casey Nicolaou from Scripps at this time, doing total enzyme synthesis, that he showed how the use of enzyme can really optimize an angioselective catalysis. So I did my PhD in the group of Iulias Monu, and there I used enzymes as an angioselective catalyst for biochemical applications. And it got us this time that we started being really excited about how can an enzyme have different specificities over time. And at this time, it was that we realized that changing a solvent or the environment, the specificity of the enzyme was changing. What was the mechanism underlying this? And this was really important because that pushed me to go to the department, to Ronald Nolte's group at Nijmegen, Netherlands. And Ronald was the group that first made the first single molecule experiments of observing in real time the function of a protein one molecule at a time. And it was at this time that we introduced the concept. We found out that enzymes do not work constantly as one would expect. They're not machines, but they operate, they fluctuate between active and inactive states, or as we call this in this review, and hopefully you can still recognize me there. It was a few years back that we introduced the concept of do enzymes sleep and work. And at the same time, we did a lot of work on the use of polymer assemblies to spatially confine biomolecular entities in distinct environments and use them as catalysts for cascade reactions. So naturally, when I came here in Denmark, I worked in the, as I said, in the group of Dimitrios Stamou, and I continued and introduced the single enzyme work in Dimitrios Stamou work, lab. But I was also involved in this establishing the single vesicle assay and then interacting with the students that were doing a lot of the liposome work in single part. So as I said, in the last 6 years, I started my own lab here in Frederiksberg campus in the Department of Chemistry. And we started end of 2015, and it was exciting that I had inherited the lab, but I had no microscopes, not even spatula. So the first months we were struggling of making any experiment. But now but now things are perfectly fine, as you will see, I think. And I will tell a few of the elements that we work with. The three major pillars of my group is adding the fourth dimension that of time in structural chemical biology for controlling cellular responses. And there are three major pillars, as I said. One of them is the dynamic structure biology, try to understand at the molecular level how the motions of the protein underlie biomolecular recognition, function, and fidelity in biomolecular recognition. And at the same time, because the function of the proteins do not exist alone in the cells, but their spatial localization, biomolecular recognition, and function will result in a cellular output and possibly at the organismal level. We started over the last years working a lot at this level. And we work with a host of protein systems. Here I displayed the CRISPR work that we did with the group of Guillermo Montoya. You probably know CRISPR as it was awarded, Emmanuel Charpentier, they were awarded by the Nobel laureate, 3 years ago for understanding how CRISPR-Cas9 works. In this paper with Guillermo, we defined how CRISPR-Cas12 works. And we use this to understand at the molecular level, and as I said, at the cellular level. And last but not least, because we're producing a wealth of data that are very hard to analyze using conventional techniques. If you want to extend beyond the state of the art, you need technologies that allow you to master the data quickly, accelerate by orders of magnitude, and free human intervention on your data analysis. And this is something that we have been developing, employing a lot of machine learning frameworks to analyze the wealth of data. Let's hope that in the coming paper, our suggestion for a cover will go through. So in the interest of time, I will only talk about two of our recent breakthrough papers in the 2021. One that is in collaboration with Amit Pandey from Switzerland and a lot of people from University of Copenhagen, and one that is mainly from our group on machine learning. So now I'm going to introduce the first pillar I'm going to discuss is about P450 oxidoreductase. And this is a protein that most of you have not heard. But what you might have heard is P450 enzymes. And if you haven't heard P450 enzymes, what you probably know is what they do. They control any metabolic cascade in our body. So cholesterol levels, testosterone levels, drug metabolism, estrogens, all hormones are synthesized and are catabolized by P450. Up to a few years ago, we thought that, and all these P450s, in order to work, they receive electrons from P450 oxidoreductase. So up to a few years ago, when there was a metabolic disorder, people were trying to find the problem on the P450s, sometimes with success, sometimes with not. But Christoph Luke and Amit Pandey, when they were in USCSF in the department of Miller, they had a paper in Nature Genetics and the Lancet later on that they actually said, wait a minute, why don't we look upstream of the P450s, the protein that actually gives them electrons and activates them. So they proposed that this time they found that mutations on the pore are actually manifesting themselves into downstream metabolic pathways that are disease related. So now this is where things become interesting, because there is a single protein and interacts with 300 P450s in plants or 50 P450s in human. So this protein has to be extremely plastic, but it also has to be extremely specific, because you don't want to activate the protein, for example, and I'm making this very simple, that synthesizes testosterone, and at the same time, the protein that breaks down testosterone, because you're running in loops. So it has to be highly specific. So how can a single protein communicate so efficiently with so many pathways downstream? And there's only one system down there that we know that does something similar, and this is G protein coupled receptors, GPCR. And Brian Kobilka was awarded by the Nobel laureate in 2012 for getting the crystal structure of GPCRs, but also defining the concept of a bias signaling. And practically what the signaling was, the bias signaling is that the GPCR, depending on its conformations, it can initiate, interact specifically, and initiate one signaling cascade, which corresponds to cell proliferation, cell division, and signaling along the cells. So hopefully what I'm going to convince you about here is a few elements. A, that POR is a completely new pharmacological target that we just discovered. B, I will introduce to you the concept of biased metabolism, which is a mechanism that is akin to bias signaling of GPCRs, but now for metabolic enzymes, a completely new way of this mechanism. And because we understand this mechanism, we're able to design and predict ligands that A, bind on board, B, bias how these pathways are connected. Practically, you just, like a maestro of a company, you just activate one or inactivate another instrument. That's what I'm going to show you, hopefully, today. So as I said, we started by identifying a few ligand molecules. So as a proof of concept, we started with doing some principal component analysis on the FDA-approved databank of molecules using the Lipinski rule of 5 and methodologies that I'm not going to go into the detail in the interest of matter of time. I'm happy to discuss afterwards. We identified a few molecules that we thought we had evidence to believe that they should be able to dock with pore and bind to pore. We went in and did docking simulations, MD simulations, and found that at least in silico, these molecules do bind on pore. Bindinger says that also we can confirm not only in silico, but that the molecules bind on pore. And then we said, okay, if they bind, do they actually affect activity? And this is a little bit challenging to observe because, as I said, pore can activate multiple different pathways. So which one do you check? So what we did is we found two of the molecules that should be activated by pore. And there's a host of data here that I'm not going to explain in detail, but what I would draw your attention is that this ligand over here activates 300% one pathway and inhibits another in vitro, of course, by 90%. So actually you have one ligand that says your protein, you go work, and it stops another protein from working. This is a completely new concept on how proteins can communicate downstream. And now the question, of course, you might ask is, all right, I can see this, but what is the mechanism? How can you find out how this is happening? To do this, we involved what we call the spectrometric ruler, single molecule thread, that reports distance information at the nanoscale. Imagine a ruler that allows you to see how nanometer motions of the proteins take place. And this is how our data look like. And I know that most of you, this looks like blinking stars, but there is practically, that's what they are. They're blinking stars. There's a wealth of information that we can analyze of this. And this is a process that involves looking at these two chromophores over there, how they blink, integrate their color, and extract distance. And it's a process that takes time, but we developed a software that allows you to transit from raw microscopy images to dynamic structural biology insights with a couple of clicks. The whole idea is based on a neural network, an advanced form of machine learning, that it integrates the data and outputs the behavior of biomolecules distance that you can extract. And it does so. The only crucial human intervention that one has to have is setting a confidence threshold of how confident you would like to be. We made a software that you can download on your Mac or your computer. It's a free standalone executable. You can find it on my lab's GitHub page over here. You can plug and play it in your computer. And this is what we call, it allows you to transit from raw microscopy images to dynamic structural biology insights with a couple of clicks. We call this one-click biology, chemical biology, because it accelerates 4 orders of magnitude data treatment while being completely free of any human intervention and potential cognitive biases. Now, by the time we analyze this behavior, what you see is the distribution of conformations that your pore has. And you can see that you can appreciate that you can see 5 distinct conformations that are on par. They are in agreement with crystal structure data that we have. And now we administrated the molecules on the single molecule thread, the ligands that I just showed, the most promising ones. And you can appreciate, and I'm not going to go through the details here, but you can appreciate that depending on the ligand you administrate, you can completely shift equilibrium. Here you can see that you put one ligand, this blue occupancy is reduced, and you put another one, and you can transit from here to here, and you have a completely new extended conformation. So practically, if I am to summarize this slide and keep it very simple and some superficial here, you can see that these ligands bind on pore, and they alter the way the protein moves around. So now the question that you might ask, okay, you showed me in vitro, in a chemical system that this works. Is this applicable in any cell biology? But before I go that, because I need to stress this, that because you're looking at the conformations of the protein, and because you're looking at the lifetimes of this transition, you can actually export the complete multi-dimensional landscape of the protein, how it some dances in space, and how this is remodeled by the regulatory cues, which in this case is ligand. So as I said, the next step was how can we see how these molecules interact in cells. So this is a fantastic and enduring collaboration that I have with a group of Amit Pandey in the University Hospital, Children's Hospital in Bern, in the Department of Metabolic Disorder. And we went there with these ligands, and we tested the effect of these ligands on the cellular level and how the CYP, the P450s, are affected within the cell. And this is a very heavy slide, but I'm only going to focus on these few elements that you can see here. What you can observe here is that some of the ligands activate 1P450 and some of the other ligands deactivate P450. And depending on the ligand that you use, you can actually almost eliminate it. I think it's also important here that this CYP17 has two functions, the lyase and the hydroxylase. One of them is involved in prostate cancer, and we can selectively inhibit one of the two functions of the same protein. This has never been achieved to the best of our knowledge before. Now, if I am to summarize this work, we introduced a completely new concept, that of biased metabolism. We found a new pharmacological target. This pharmacological target can be targeted by new ligands that we can express and produce and test. Ligand binding alters the conformation of protein, and by this affects the conformation of the protein, affects pathway activation in humans. It's a completely new paradigm, and the way social media and press media in Denmark and newspapers in Denmark discuss it is the conductor of your hormonal orchestra. The way your molecule moves around, it activates different pathways in cells. And the way we did it is because we have a profound understanding of the energy landscape of the protein, the mechanistic insight, as I told you, and your ligand stabilizes certain conformations, each of which are linked to a distinct pathway. And the beauty of this technique is that now you can design ligands that do not kill pore activity. If you stop completely pore activity, it's lethal for any organism, but it can bias its specificity, so you can make one pathway versus another. So you can eliminate deleterious pathways and enhance good pathways in the human. So that concludes the first part here of the work. And this shows how understanding at the protein level, you can control a response at the protein, but also at the cellular organism. The second part of my talk is the implementation of how the spatial localization of proteins and their function will control output at the cellular level. And I'm going to introduce you the concept of single particle tracking, and this is a nanoscale object that we can track using a microscopy and see in real time how it moves inside the cells. Now, the advent of single particle tracking techniques have provided a wealth of information. Okay, I'm going to stop the movie because then everybody just looks at the movie and not listening. So it provides a wealth of information about all biological process, that is, for example, cellular endocytosis, drug delivery, viral entry in the cell. And you can appreciate when you saw this movie or when you see some of our data that this behavior is highly heterogeneous. So what you can see here are lipases, and I will come back to you, as they degrade fat. This is in collaboration with Novozymes. You can appreciate all of them showing slightly different behavior. You can track how nanocarriers loaded with pharmaceutics penetrate the mucosal barrier and deliver in cells in real time, or how insulin, this last one is in collaboration with Knun Jessen, how insulin receptor loaded with insulin, different forms of insulin, internalizes inside the cell, an important element if you want to battle insulin-related diseases. So how do we do this analysis? We use advanced microscopy, and probably to this audience, I don't need to introduce how the microscopy works, but the whole concept is that you take consecutive images of the same area, and you see these white spots over there. You localize this white, you identify the elements, you localize them with diffraction limited resolution and extract their position. And then what you do is you link the particles together. So when you saw the lipase moving around like this on the surface, this is the track, as we call it. This is the pathway that the enzyme followed. By looking at this track, you can appreciate that it's highly heterogeneous. You have something that is almost linear here, then the protein here just completely trapped in something, then it moves slower here. And this heterogeneity now underlies function at the molecular level or at the cellular level. but also imposes a considerable challenge to the analysis because you are acquiring thousands or even millions of traces like this, which are very hard to analyze manually. So the first step is like, how can we define a particle? What is a particle and what is not a particle? This is work of a fantastic student of mine that I'm so excited to see that he woke up in the middle of the night and see this, Henrik Pinhold, that defined the particles using a machine learning framework, all the elements that define the particle. And by using the machine learning framework, you can clearly set, instead of setting a single threshold on intensity, you can identify all the features that define the particle and select the particle and separate this from the background. And we can do this with a high fidelity, more than 99% accuracy. And by the time you analyze this behavior, you can extract completely new insights about how proteins work. Now, this is work that we do with Novozymes explicitly. And most of you, when you hear the word protein, you expect this to be a molecular machinery, a robot that catalyzes continuously a chemical reaction. As I will show you, proteins do not work continuously as we want them, but they actually have sleeping hours, working hours, and when they work, they don't always work with the same working efficiency. Now, if you are an employee and you want the job of your students in your research group, or if you are in a company, the job to be done, you want people when they are in the office to work flat out and no breaking and no working hours. Now, if you're an enzyme manufacturer, you want your proteins not to have working hours and working efficiency. You want your proteins to work 100% of the time. We cannot really do it with students here. That would be highly uncomfortable for most of us. But in a perspective of the company, this is fundamental. So how can we find this and how can we tailor this? So I'm presenting to you a methodology that was developed by Soren Bohr in my lab. And this is probably the smallest and yet the most hideously expensive washing machine that you can make. This is a glass slide that we can put on the microscope. On this, we have a fat layer that constitutes, which is made out of lard. And this would be like a stain that you have in your clothes. And you add the washing powder, the enzymes that Novozymes is producing to degrade this washing powder. You add the enzymes, you label the enzymes with a chromophore, and you add them on the surface. And then you, using TIRF microscopy, you can image these enzymes as they degrade the fat. You can, now what you see over here, each black spot corresponds to a single enzyme, 3 nanometers enzyme, degrading fat. You can clearly appreciate that they are highly, highly heterogeneous. There are guys that are stalling. You can see my mouse for a while, then they start working. They wake up and they start working very fast. And then they get tired and they may stop for a coffee somewhere. And then they work again. You can see other proteins over here. This fellow over here. This guy is enjoying his mojito cocktail on the sunny beach of the south and does nothing. Now, as I said, if you are a company, the only thing that you don't want is your employees to drink no, if you are a manufacturer of proteins, you want your proteins to work flat out and not stall. And now, how can you gain this layer of information? This methodology that I'm presenting to you, when we do it for active and inactive proteins, we can clearly see that the faster they work, the fastest they move. If they don't work, they're practically immobile. If they work, they're propelled by substrate and they move 2 orders of magnitude faster. So it's a direct correlation of activity to displacement function. And by doing this, and I'm now going very superficially, we can actually plot here the speed versus time of each of the proteins that you see over here. And just to put things into perspective, in a single day, we can acquire 50,000 of these proteins. In a week, 1 million of them. So even though you're looking at the single molecule level, you reach values that are applicable to an experiment in a test tube, but yet with the unprecedented fidelity of single molecule studies. So what you can see here is that this protein, for a fraction of its time, is completely immobile. It's actually sleeping, it's not working. And in the beginning, it was working very fast, and then it got tired, it had a nap, and then it woke up and it worked again. So, and now you can do this, obviously, if you were measuring something in bulk, you could completely average this information, but the single molecule results reveal this information. And then you can go out and see what is the fraction of the time that the enzyme was spending working hard or working slowly, and what's the fraction of the time they spent on the target, on the stain of your clothes. Needless to say that all this information, if we were doing a bulk assay in a washing powder, would be completely averaged out. So you see a protein works, it's more efficient, but you don't know if the protein is more efficient because it spends 20 hours in the office, or it only works 8 hours a day, but it works extremely fast. But by the power of the methodology that I just explained to you, you can actually delineate these elements and actually start pinpointing what are the mutations you need to do on a protein to make them more efficient. This is work that we do for many years with Novozymes, and I'm happy that I continue this also with a professor from Crete, Kelly Velonia and Erica, that we make biohybrids that we worked with Novozymes on making novel biocatalysts. Now, as I said, this behavior of the protein is highly heterogeneous. Now you can see here, you can appreciate that some of this is linear, then confined, and then it's stopped. How can we extract this layer of information? Now, if I am to analyze very specifically this, very generically this, the types of motions a molecule will have is what we call a normal motion, directed if it's pushed, confined in a small area, or subdiffusing. And I know this sounds very, very tricky to you, but you can imagine that you see a leaf falling down from a tree. This part of the leaf, okay, maybe that's not entirely correct, but this, if a molecule is moved by the air, it's probably going to have what we call a normal diffusion mode. If it's directed, because it's in a car, for example, then it's called directed. If you are in a small area, in a small office, it's confined, and so on. And these motions, this type of motions, underlie biological response, because depending on what they are, it means that a protein in the cell, it moves in a certain. And how do we analyze this? Classically, we analyze them using the mean square displacement. So we take all of them, and I'm being very generic, and you say, oh, all of your molecules move with a speed of this micrometers per second, and this is if they are directed or not. It is like saying the average speed of cars in Copenhagen is 20 kilometers an hour. It almost provides no information if cars are moving on the highway or if moving on side streets, etc. And this is where Henrik, an extremely talented student who is now in MIT doing a PhD, went in and said, I'm going to do much better than this. So he developed what we call a very generic method for the analysis, processing, and classification of single molecule data using what we call a diffusional fingerprint. And this is based on a machine learning framework. And I'm going to try to make very simple and very heavy slide. He went in and identified all the features that describe the behavior of an enzyme. How fast does it move? Does it move on a straight line? Does it make turns? Etc. etc. Then the method operates by ranking these features, outputting the ones that are most important, and predicts behavior based on this. When it does so, it creates initially a 17-dimensional space. Here, this is collapsed on the 3 dimensions of the most important features. And I know this sounds very techy, and I will make it extremely simple and explain this. You're sitting in the main corridor of HSU, and you see a person walking, and you say, huh, this person is suspicious. Why is this person suspicious? Is it the way this person dresses? Is it the clothes? Is it the color of his skin? By the way, I mean no anything but to any skin color or male, female. Please don't take this in any wrong way. Or is this person running around with a laptop in his hand? What are the elements that trigger you to think that this person is suspicious? And then the next day, you see another person, and you say, oh, this person is suspicious. But it could be completely different features that define you to allow you to believe that this person is suspicious. So how can you classify this? And this is what Henrik did. This is what the machine learning framework does. It takes all the features, it ranks them with significance, and outputs the behavior and the contribution of each one of them to classify a distinct behavior. Again, this is a software that is freely available on my GitHub page. It's not a software, it's a script on Python. It's open access, freely available on my web page if you want to record this. And by doing this, Henny can go out and just by looking at the behavior of a protein, output the characteristics of the protein. Either this is a mutant of a protein, or this is a protein that operates, is regulated by calcium, by detergent. Here I show some lipase that we do with Novozymes. And you can see just by looking at the behavior in a model-free system that trains and predicts on experimental data, you can output a specific behavior. A fundamentally new insight on how do we characterize biomolecular behavior. And now I'm showing you data on lipases and how we can simply classify by the way people move around, lipases move around their behavior, but this is applicable to a host of biological systems. So we tested this on nanoparticles loaded with pharmaceutics penetrating the mucous surface, enriching and delivering pharmaceutics cells, transcription factor diffusing on DNA, fundamentally important for cancer, but also lipases on different substrate surfaces as well. And in all cases, not only the model predicts with often 90% accuracy just by looking at the traces, but it also outputs a mechanistic insight. It says, oh, this is a nanoparticle that will deliver more efficient because the mean step length is this. So it outputs a mechanistic insight. And as I said, this is applicable to any system. And we expect, and we are testing this, we expect this to be able to predict, for example, if a receptor is moving on the cell monomer as a dimer with or without ligand, as we showed you for the transcription factor, or allow us to exemplify how topographical or geometrical variations in the diffusion medium will allow a separation of the molecules. And this is what we do in the last 2 minutes, because I also need to keep track of time. I'm going to show you some of our very recent work on a very well-known and deleterious virus that we have been dealing with over the last year. So this is in collaboration with both Hannemon Nielsen and Jörn Kemps and also Tom Kirkhausen. And this is, what you see here is VLP particles, virus-like particles, scouting and trying to find the weak spot for penetrating the cells and delivering the genetic material in the cells. The lines that you see here correspond to individual tracks, and I'm going to play this a little bit slower for you. And you can see that they're scouting the surface of the cell. And if you see here, by the time they find the correct endocytotic pathway, they will bullet through and penetrate the cellular barrier, be directed in certain cellular compartment, and deliver. My movie doesn't play properly, and deliver their cargo. And if you look throughout the cell, not only you can see that multiple cell entry pathways are happening at the same time. So how can you analyze this data? And how can you extract quantitative information? And this is where Jakob comes in. And he said, if we are to analyze 100,000, how is conventionally be done? Conventional people would take this trajectory, which is the spatiotemporal localization that I showed, and say, oh, this must be on the membrane because it moves slower. This must be, for example, on the endosome. So if I cut this part, then I know how much time it spent on the membrane. This is in the endosome. This is where it releases. And of course, you can do that. But if you want to analyze 500,000 traces, you need 100 PhD to do that. And Jakob is not one of them, because he said, I am very much smarter than this. I'm not going to spend my time on this. I'm going to spend my time programming a machine learning framework that is going to do this. So, as I said, conventionally, people go and manually annotate this. And what Jakob did is he developed a machine learning framework that goes out and takes this entire trace and says, for this part of the time, my molecule is subdiffusing. And for this time of the time is directed, and right there it now becomes normal. And not only that, but he trained his neural network to output not only what the track is, but also the confidence of outputting this prediction. So as you can see here, it is very confident that this is a subdiffusing behavior. Very confident that this is directed, and here it's like, maybe it's normal, 20%, but then it's like, no, it's directed, and then it becomes normal. So it also gives you a confidence interval of your predictions. And not only that, but by the time you segment your traces, you can do the fingerprinting that Henrik developed in my lab, and then you can output on top of this, what is the fraction of the time that your molecule was doing this motion? For example, how long did it stay on the membrane? How fast was it moving on the membrane? And when it left the membrane, where did it go? How long did it stay there? And how fast did it go there? And the beauty of this is that you can analyze terabytes of data in a few minutes, a process that our collaborators, Tom Kirchhausen, need 2 months to do in Harvard. And Jakob is going there in 2.5 weeks to implement this network on other viral entry. And I'm not going to bug you with the details, but by the time you do this for all different viral entities that I just described, for the wild type and the delta and the omicron, you can identify the distinct internalization pathways. And by the time you have the distinct pathways, you can see in real time which of the molecules you can use, inhibitors of each pathway, to stop the virus enter the cell in the infection. Obviously, I cannot indulge into further details because we're about to submit this paper. Hopefully you will see it on bioRxiv soon, and I can provide much more details. And at this time, I need to thank a group of people that actually do everything. I pretend, as I said, to give them directions, but they actually usually tell me what to do. It's a fantastic group of people. We are constantly expanding. We need to hire 3 postdocs and 3 PhDs in the next 6 months. So stay tuned. The 2 postdoc positions are already open, and the PhDs are coming up. And I talked explicitly about the work of Soren on the single particle, Henrik on the diffusional fingerprinting, Simon on the biased metabolism, and on the virus is Frank with Emily, and the machine learning by Jakob.